Hello, everybody. Welcome back. Today I'm alone because, as you may know, it's harvesting season for some crops, and so both Philippe and Claire are busy in the field. But today the screen is going to be very crowded because we have many amazing speakers. And so, as you know, we always start with a brief introduction about PhenomeForce, and then I'm going to get the speakers on. So, who we are. So, for the people that they know us. Thanks for being patient for this repetitive introduction. But for who is new, so PhenomeForce is a platform that we started about a year ago, which aim to provide free training and opportunities for early career researchers in phenomics. But our main goal is to bring people working on phenomics together so they can interact and they can share knowledge. So we have been quite successful, and so we thank all the people that keep following us, and we hope to reach out to more people in the future. So this project is, I'm Anarita Marrano, I'm a postdoc researcher from University of Massachusetts, and I started this project with Felipe Matias, which is currently from North Dakota State University. And this year we also have Claire Gann from the Agriculture and Agri-Food in Canada. And also we have been strongly supported and helped from Noah Falgren. So how you can reach us and visit us. So we have a website where you can find all the information that we, all the events and the resources that we share. You can also follow us on Twitter, where we usually post news. We have a YouTube channel where you are currently watching us, and so if you want to be updated about our workshop or our resources, please subscribe to our channel so you will have notification. We have also an email, so you can reach out to phenomeforce@gmail.com. And this email is also important because if you want to join our Slack workplace, you can just send us an email and we will add you to the channel. So for whom is not very familiar with Slack, Slack is a platform that allows people to connect and communicate. So we created this Slack channel about a year ago. It's starting to be very useful for some people and to connect people and talk about topics. So here there are the channel that we created based on the topics in phenomics. You can join any of them. If you join the Slack channel, we ask you to introduce yourself in the introduction channel. But if you have any suggestion about this workshop series or anything else, you can reach out, you can write in the suggestion channel. So as you can see on this YouTube, we have been we did the first workshop series of Friday Hands-On last year. We had 10 amazing workshops, and you can rewatch them anytime at our YouTube channel. And also the workshop that we are going to record today, you can always watch it again anytime in this platform. So this workshop series of 2021 focus mostly on low-cost phenotyping platform, but also on data analysis. And so we are trying to provide still resources that can be useful for any kind of projects on phenomics. So usually the workshop have a short introduction, and there is a hands-on section. This is the 8th workshop. There will be other 2 for the next 2 Fridays, and then also this workshop series will end, but we are already thinking about next events. And so talking about the workshop, today we are going to talk about PhytoOracle, which is an amazing pipeline used mostly that can be used for analyzing data from many platforms, from many sensors, such as drones. So these links are also available in the description of the YouTube of this YouTube video, but also I just posted in the comments a link that can give you access to the GitHub page where there are information about the hands-on section of today. And so, speaking about today, I'm going to introduce the speakers of today. They are part of the research group of Duke Pauli, but today we are going to have Emanuele Gonzalez, Arian Zarei, Travis Simmons, and Michele Cosi. And they are going to talk about PhytoOracle and how we can use this pipeline. And so speaking of them, I'm going to add them to the screen. So welcome Emmanuel, welcome Travis, welcome Arian, and welcome Michele. So you guys are muted. So it's going to be very fun. I want also to say that, as you know, we are online, and so if there are any problems with internet connection, please stay there. We will try to solve and to keep going. So also, if you have any question about the pipeline, you can write on the YouTube comments. And so I think Arian is going to manage the question today. Thanks for helping me. But of course, so they will try to answer the question or on the YouTube, or we will stop the speaker to answer live on the YouTube. And so saying that, I will stop in sharing my screen. And I think Emmanuel is going to be the first speaker. And so if you, Manuel, wants to share your screen, I'm going to put you on the live. So, okay, so Emmanuel, you are going to be the first. I'm going to share the screen. So I'm going to leave you alone leading the screen. And so if, of course, when all the others need to be put on, please let me know and I will work on the background. Perfect. So good luck, guys.
Can everyone hear me before we get started?
Yeah, we can hear you. Very good. So good luck and enjoy.
All right. Good morning, everyone, and thank you for being here today. As Anarita just mentioned, we will be talking about PhytoOracle, which is a series of scalable and distributed workflow pipelines aimed at analyzing high-throughput phenotyping. Now, these pipelines were developed to process data from the University of Arizona gantry, also known as the Field Scannalyzer. But these pipelines can also be used for other platforms such as drones, indoor plant phenotyping platforms, and satellite imagery. Now, one of the reasons why we developed these pipelines was because as sensors become cheaper and faster, distributed workflows such as PhytoOracle will be required to extract actionable data from large image datasets. And this will in turn enable research aimed at connecting phenotypes to genotypes. So to begin, I would like to just give you guys a brief overview of our pipelines. We have a series of different data sets ranging from stereotop RGB, FLIR thermal sensors, PS2 chlorophyll fluorescence, and a scanner 3D sensor as well. Now, all of these data are processed in a scalable fashion through the use of a tool known as CC Tools, which was developed by a team at the University of Notre Dame. Now, this tool allows us to create a master worker framework for distributed computing. And this works not only on high-performance computers, also known as HPCs, but also cloud resources. And this allows us to process each one of these data sets that you can see here on my screen. Now, this framework allows us to process thousands of jobs in parallel on nearly any Linux environment, which significantly expedites the processing of phenomic data. So with that in mind, let's go ahead and get started by following the workshop preparation. Now, for those of you that have clicked the link that Anarita sent, just make sure that under the branch, you're under the PhenomeForce branch under our GitHub repository. And that's where you'll see this document here. So now if I click on create a Cyberse account, the first bullet here, that will take us to a Cyverse webpage that will allow us to sign up for Cyverse. So if you haven't done so yet, go ahead and click on Sign Up and fill out these 3 different sections. It'll ask you for your name, your affiliation, and last but not least, some more information about your research. Now we're going to pause here, give everyone a second to fill this out. And if there are any questions, please feel free to post them in the comments. And Arian will be in charge of letting me know once everyone is done. So whenever you have created your account or once you're logged into your Cypress account, go ahead and just comment done in the comment section. And if there are any questions, also feel free to post them in the comments as well. We're here to help in any step.
Oh, there is an echo. Yeah, so thank you for doing this. I think someone say done. So yeah.
All right, so we can move forward. For those of you that need more help, feel free to comment in the section if I'm moving too quickly as we carry on. So once you have your Cypress account, we're going to click the second bullet point here to register for our workshop today. So let's go ahead and open that link there. And it'll ask you to log in. So we're going to go ahead and do that. And once we log in, you'll see our PhytoOracle workshop. Now here, I'm already enrolled for this workshop, so it's gray in my scenario, but for you guys, it should be blue and it should say enroll. If you click on that, it'll enroll you for our workshop today. And McKelly will be in charge of approving that. And for now, we're going to go ahead and continue with the next steps. So I'll pause here just so you guys can enroll, and McKelly will give the approval.
Emmanuel, can I ask a question just because? So if, for example, in the future, a person is going to watch the YouTube and is not going to watch it now, so will this be still available as opportunity to redo the workshop later on?
Yes, most definitely. We will have this up and running for the next few days, and we'll try to extend that as much as possible.
Okay, thank you.
Okay, so once you guys have registered, you should see under the services section, the atmosphere link. So we're then going to click there. And this will take us to our Cypress dashboard. This is where we can request instances, we can set up different projects, and this dashboard here will tell you how many resources are allocated to you. For this workshop, we'll have more than enough resources to do our data processing. So for now, we're going to go ahead and create a new instance. So we can click on Launch a New Instance. And what we're going to do here, we're going to then search for phenome_force. And we'll click here. And this is our virtual machine that we're going to launch today. So up at the top right here, we're going to go ahead and click on Launch. And this will bring up a menu to launch our instance. So here, everything else is already pre-selected for you. The next step here is going to this instance size section, and we're going to change this from tiny 1 to medium 2. Now, all of this information is found here. We also have screenshots for you to follow along if you decide to do this workshop later. So again, we're going to choose the base image version of 3.0, and we're going to choose an instance size of medium 2. Now, once we have done that, we're going to go ahead and launch the instance. So I will go back to that tab and click on Launch Instance. So the next step is it's going to start building this virtual machine, and it might take 10 to 15 minutes. So in that time that we wait, we're going to go ahead and pass it over to Travis Simmons, who will get us started in explaining what our PhytoOracle pipelines are and why they are likely of interest to you all. So Anarita, if we can now hand it over to Travis Simmons.
Hello, everyone. Let's see, get my stuff shared here. All right, can everyone see that okay?
Yeah, we can see it. Thank you,
Good
Travis.
deal. All right, so again, hello everyone, welcome. Thank you for joining. And so around 6,000 years ago, the people of Mexico and Central America started domesticating teosinte by selectively breeding for desirable traits, setting the stage for modern corn. These traits could be a whole host of things, such as plant height and the quantity or quality of yield. And back then, these traits would have been measured by hand. As time passed, though, we moved to more efficient trait measurement tactics, such as drones, phenotyping carts, and outdoor phenotyping platforms, giving rise to the field of phenomics. The vehicles with which we collect data are not the only thing that has changed over time, though. Advancements in genetics have allowed us to understand that phenotypes are a function of not only genes and gene combinations, but the environment in which the plant is grown. This genotype-environment interaction that gives rise to phenotypes is what our team is interested in, just as I'm sure many of you are. So in order to do this, we use the Gantry Scanalyzer, which is the world's largest phenotyping robot located in Maricopa Agricultural Center in Maricopa, Arizona. The Scanalyzer is equipped with 5 different sensors, each of which inform us about different phenotypes being expressed in our plants. With the first 2 sensors, we can get very visual and morphological data, such as with our 3D line scanner, we have been able to gather plant height and volume measurements, and we hope to capture more traits such as leaf morphology. And from our RGB sensor, we can extract bounding area and canopy cover. However, the last few are a bit less visual and arguably more fun. So from our hyperspectral camera, they can analyze more than 900 bands per pixel and extract physiological data such as solar-induced fluorescence and disease and water content, all kind of fun stuff. With our PS2 fluorescence sensor, we can quantify the plant's FV over FM, which is a measure of the plant's photosynthetic efficiency. And with our thermal sensor, we measure our plant's transpiration and thereby its water use efficiency. So we can run these sensors and collect all this informative data about the phenotypes being expressed in our plants and inform breeders about top performing lines. But this data is bulky, with the maximum output of data per day coming in around 10 terabytes. And this is the real focus of our presentation. Because although not everyone is using a gantry for the phenotypic research, this massive influx of data is a problem that we're all facing and what we hope to address today. Now, I'm going to hand it off to Michele to talk a little more about our novel system-agnostic approach to handling this massive amount of data.
Hi, good morning, everyone. Good afternoon or good evening, wherever you are. I'll be taking over for the second part of this presentation. And thank you very much, Travis, for starting this up. And thank you very much, everyone else in the team. So as Travis mentioned, this is a lot of data being generated, and we not only have to start thinking about how to extract these phenotypes, but we also have to think about how to handle this data. And this is why we created PhytoOracle. PhytoOracle is not only the name of our team, but it's the name of the pipelines that we have created in order to extract these phenotypes from the images that are collected from the gantry analyzer. And PhytoOracle, all of the pipelines in PhytoOracle follow the same structure. So we have raw images that are obtained from the gantry analyzer. And one of the first things that we do is to geo-correct and create an orthomosaic with these images. So from these single images, we are able to create a field orthomosaic from where we can then clip single plots and finally extract information, valuable information that we can use for analysis later down the line. And in this example right here, we have a FLIR camera, which is our thermal camera from which we can extract temperature information at both the plot and plant level. We can do this for PS2, as we're going to be seeing today, as well as RGB, and recreate 3D point clouds using our 3D sensor. So next slide, please. We talked about data and the size of the data, and addressing this has been quite challenging, but is one of the things that we all need to think about as phenomics in the field of phenomics. So what the flow of data looks like for us is what I have here in the slide. We have data generated from the gantry analyzer, which is then stored in a cache server up to 3 days. From the cache server, the data is then transferred to the Cyberse data store, where it is which is going to be storing not only the final data set, but also all of the raw data that was generated by the gantry analyzer. And the Cyberse data store, I mentioned that it holds both types of data, and in order to process all of this data, we use the University of Arizona high-performance computer. And the way that we have it set up is that all of the data is transferred to well, not all of the data, but the data is transferred to the master node, which is part of a manager and worker framework, which allows to parallelize all of this job, thereby scaling processing upwards and reducing the overall processing times. This manager-worker framework, it's allowed by a software called WorkQ. And WorkQ, which is part of the CC Tools suite developed by the University of Notre Dame. And this parallelization allows for very quick generation, extraction of our phenotypes from the worker nodes to the manager node, which then compresses everything and sends it back to Cyberse. The users can then not only access the final data set, but also all the other intermediates from the Cyberse data store. So I want to put a number to it. And let's ask a question. How much time would it take to process a single season worth of RGB data, which is roughly 50 terabytes, on a 4-core regular lab computer? We have estimated that it will take roughly 55 years. And that's an insane amount of time. So how much does PhytoOracle, which, keeping in mind, works on a HPC, on the University of Arizona HPC, how much time would it take PhytoOracle to process all of this data? Roughly 6 days. And when you think about it, that's crazy. We process this allows us to process data very, very quickly. And this is what we want to show you today. PhytoOracle is not only a phenotype extraction platform, but it's also an automated platform. Here on the left, we have an example of how it works. These are Slack messages. If any of you uses Slack, I'm pretty sure you're very familiar with what the messages look like. But these are Slack messages that are sent to us whilst PhytoOracle is working. And we can see when the data is being downloaded. We can see when the data is being extracted. We can see when the data is being compressed, sent back to the data store. And it's very useful when it comes to understanding at what point the pipeline is at. And here on the right, we have two examples of what the data extraction looks like. On the top, we have our thermal data. And this is a season-long experiment where we compare well-watered plants against water-limited plants. Well-watered in blue and water-limited in orange. In this experiment, we wanted to see the temperature of the plants when they are under drought and the difference between well-watered and water-limited. And what we were able to observe is that when the plant is under drought stress, they tend to have a higher temperature overall, and this is what we observed throughout the season. We can use this as a proxy to understand stomatal conductance. For example, why are the plants that are under drought stress retaining more water? Well, that's probably because they have a lower transpiration rate. And that is something that is a sign of drought stress. Here on the bottom, we have an example for photochemical efficiency, which is calculated using FV over FM, FV being fluorescence variance over FM being fluorescence maximum. And we also have water limited in orange and well watered in blue. And throughout our season, this is also what we observed, that when the plants are under stress, they have an overall lower photochemical efficiency when compared to well-watered plants. And this is also another sign of plant responding to drought stress, which was expected. Thereby, so if we go to the next slide, this is what we wanted to introduce you to today. All right, so we wanted to introduce you to what the data looks like and how the PhytoOracle pipeline works. And in today's tutorial, we want to allow you to mimic the PhytoOracle pipeline using the Cyberse virtual machines. Although we cannot allow you to do this on a HPC, we can allow you to use our virtual machines so that you can understand and have a grasp on what is going on and how to run the pipeline. And we are going to be using thermal and our photochemical efficiency data, which I just showed you. And then we're going to be analyzing this data using the Jupyter notebooks. So now I am going to hand it back to Emmanuel, which is going to carry us through the second part of the tutorial.
Okay, everyone. So now we're going to, there we go. Okay, perfect. So now we're going to go back to the document that we were originally on and continue with our workshop. So just to remind everyone, we went ahead and deployed a virtual machine that was a medium 2 machine with 4 CPUs, 16 gigabytes of memory, and 160 gigabytes of disk space. Now, the next step is to wait until our virtual machine shows a status of active. In this particular case, we are still deploying. So for now, what I will do is walk you through some of the information here as it relates to virtual machines and how we usually run our pipelines. So as Michele mentioned, we usually run our pipelines on a high-performance computer. And although we run our pipelines on a high-performance computer here at the University of Arizona, most HPC systems are quite alike. So basically, they run off of a Linux operating system. And what we do is we use Singularity images at each step. So we take Python scripts that we use to process each one of our individual data sets, and we do something known as containerization, which is using either Docker or Singularity. So once we have our scripts ready, they have been validated, we create containers that we then deploy on this Linux-based high-performance computer. Now, today, we did not use a high-performance computer. We are instead using a virtual machine just to show you guys that this is deployable on any Linux-based system. So this particular Linux-based system is an Ubuntu virtual machine. And within this Ubuntu virtual machine, we have pre-installed our GitHub repository. We have pre-downloaded some data for you guys to process. And we have also made it easy for you guys to download each one of those Singularity images. So now, once we have those Singularity images, that is what then gets deployed using the CC Tools worker framework. And as you guys can probably imagine, these virtual machines are quite useful because it allows you to process not only large amounts of data like what we use on a daily basis, but also drone data. And that's kind of where most people are at the moment, is using drone technology. There are also a lot of indoor plant phenotyping platforms. So because of this, we wanted to make our pipelines work for that diverse array of data. In our case, as I have shown here in the documentation, we have RGB sensors, thermal sensors, PS2, and scanner 3D top data. Now, each one of these may be specific to the gantry, but say, for example, you collect images with a drone, you can process your data using PhytoOracle. Likewise, if you have a thermal camera, say, for example, on your drone or in an indoor plant phenotyping system, you can also process your data using PhytoOracle. And I know that a lot of indoor plant phenotyping platforms have PS2 chlorophyll fluorescence sensors as well as a lot of remote sensing applications are dependent on scanner data, for example, 3D line scanners, among others. So just keep that in mind as we go along. Although we are processing the gantry or the field scanalizer data, these pipelines and workflows are available to you guys and can process other types of data as well. Okay. So we will just give it a few more seconds to finish deploying. So what I would like to do now is, for those of you in the live audience, if you have any questions, go ahead and post them there. And Arian can go ahead and alert me if there are any questions. Or if you don't have questions and you want to comment thus far, feel free to do so as well. And it would also be interesting to know what type of phenomic data you all are processing. So feel free to post anything in the chat while we wait.
I think a good question that I will say is, like, maybe you guys can talk a bit about Cyvers for the people that don't know what is it, because I know a bit of it. But yeah, so like a server that I think it's a public server that people can use it also for other purposes. So while we wait, I think it can be a good, people can know, because I think it's more popular here in the United States, but I don't know if abroad they know it. So maybe it can be useful.
Definitely. So Cyvers allows the use of compute resources. Like, for example, here we are launching virtual machines. It also provides storage capacity for research. So for example, McKelly mentioned that our field seasons are usually about 20 terabytes, if not more. So Cyvers provides a system not only to store, which is called the Cyvers data store, but also a system to allow you to access your data through the command line. And this becomes very useful with processing pipelines such as PhytoOracle, where most of your work is being ran on high-performance computers that are all terminal-based. And compared to, for example, a regular download off of a web page, it is significantly faster to use the Cyverse iRods system. So they provide that as well as other workshops that are available. They have a webinar series as well. And in general, they just help research advance by providing these computational resources as well as training. So now, hopefully that, Anahita, that was in enough detail for the audience. If you guys have more information, always feel free to reach out to us. Within our GitHub, you can find our email. So I will scroll down to the bottom here. You will find our email here, which is phytooracle@gmail.com. So feel free to always send any questions that you may have, if they're related to PhytoOracle specifically, or Cyverse in general. So now our virtual machine is up, so we will continue on to the next section of our workshop. So I'm going to go ahead and scroll back up here, and you'll notice here's a screenshot showing indicating for us to wait for the status to change to active. So you have to make sure that your status is not only active, but that you see a green dot right next to your virtual machine. So now what we're going to do is, once you see that, we will click on the PhenomeForce PhytoOracle VM, and we'll scroll to the bottom. Now down here, you will see this open web shell. If we click there, you will see a terminal window open up on your web browser. This might take a little bit of time, but not too long. There we go. So now this is indicating that we have connected to our virtual machine and that we are ready to proceed with the rest of the workshop. Now, I have also added more information for those of you that are more familiar with the terminal. You can go ahead and also connect to your virtual machine using your terminal installed on your computer. So now let's go ahead and prepare for processing our data. So to start off, we're going to go under the pipeline preparation section. And what we're going to start off doing is moving all the workshop contents and then changing directory into that particular place where we move our contents to. So if you hover over the command, you'll see this copy button. We're going to go ahead and click there. And then on your terminal window, you can right-click your mouse and it'll paste your command in there. Alternatively, you can use Control-Shift-V. And since I'm familiar with the terminal, I'm going to go ahead and open up my terminal and connect using that, just to show everyone how you would do that. So the way that you would do that would be SSH, your username, in my case, it is as such. And then you're going to copy this IP address here under the copy button, and then just paste that into your terminal. So we're going to do that. And then it'll ask us for our password. Just make sure that you type in your correct password, and it'll give you a few attempts just in case if you place it incorrectly. So as you probably noticed, it won't actually tell you that you're typing, but it will be actually inputting your keyboard strokes into the terminal. Okay. So now that we have connected to our virtual machine, we can continue with the copying and pasting our commands. So I went ahead and copied that command, and now I'm going to paste that into my terminal. And we'll give it a few seconds, and now that command has completed. And now we're going to copy and paste this second command, which is going to prepare our pipeline, and it's going to download all of those Singularity images that I was referring to. So just to remind everyone, these Singularity images are comprised of a Python script running within a container. And a container is essentially a contained environment. I like to think of it almost like a tiny computer that is independent of the actual operating system. And this allows you to basically bundle not only your processing scripts, but also all of the necessary dependencies to make your code run. So instead of handing over a Python script or an R script to one of your colleagues or one of your collaborators, instead, you can create a container that can be run with a simple command. So in this case, we are downloading the Singularity images for both our PS2 pipeline as well as our FLIR thermal pipeline. So this is at about 65%. And then what's going to happen afterwards is it's going to uncompress this data, and then we will be able to continue with our workshop. But for now, I'd like to direct you over to some more information that we have provided here within our GitHub repo. We have more detailed documentation with instruction manuals and examples, and here's a link to that if you would like to follow that after this workshop. We have also provided more information on all of the supported sensors and other pipelines that we won't be showing today. And the reason why we can't show RGB or scanner 3D top is because these pipelines usually take a little bit longer to process. So in order to make this workshop accessible and as user-friendly as possible, we have decided to use thermal and PS2. But just know that those pipelines are also available for your use if you decide to do so. Now, we have also provided all of the code that is running within our containers, as well as our Docker Hub repositories. Now, Docker Hub allows you to not only store your containers for later use, but also share them with others. Okay, so we are just about ready to go. As you can see here, we are now uncompressing these files, and they are a variety of different .simg files. And what that represents is a .singularity image file. So these are Singularity containers. Each one of these represents a very specific workflow that we will be running. So what I will do now is go over to our FLIR thermal pipeline just to give you guys a sense of what each one of these containers is doing. So if we click on, oops, I apologize, I did not point it out correctly. If we click on the FLIR thermal data link here, it will then take us over to our documentation for that particular pipeline. Now here we have included a figure to show you what exactly is going on at each particular step. So for this data, what we're doing is we're downloading raw data and then compressing it. We are then doing an image conversion to a GeoTIFF or a geo-referenced TIFF image. And then we are orthomosaicing those images, clipping to the agricultural plots, and then last but not least, detecting each individual plant. Now, the way that we do this is through the use of machine learning algorithms. Now, you will see once we process these data that this plant detection is carried on using a faster RCNN detection network. Now, this detection network was trained on thousands of images coming off of the gantry, and they were hand-labeled by the team that you see here today. Now, what this allows you to do is to detect each individual plant in your field. And we also have these trained models available on our GitHub repository for your use. Now, we have trained models that range from lettuce to sorghum, and we are also planning on doing some more crops, and we'll go into that later. But for now, we have some more information here about each particular container that is running. And just so everyone's aware, we have what we will be processing today is a pair of metadata file and one image file for our thermal data. And each folder contains one metadata file and one compressed image for this particular pipeline. But as we will see, this changes depending on the data set that we will be processing. So for now, since everything is ready to go, let's go ahead and now go to our setup guide here. And underneath, we will see this running PhytoOracle on an Atmosphere VM. So we're going to hover over and copy this command and paste that into our terminal. Okay. So then what we're going to do next is actually run our pipeline. So we have set up a shell script that has to be provided the date that we are processing. And before we do this, I would actually like to show you guys what the contents of our directory are. So here we can see this scan date, which was 2020-08-21. This is what contains our images. So for example, if we list the contents of this directory, we can see that there are a variety of different subdirectories. So now, if we ls each subdirectory, we can see the contents. And just a heads up, this might get a little messy. So you can see we have all of these bin files and complementary metadata JSON files. So now that we have a sense of the data itself, we can see all of these different shell scripts that are running within our pipeline. And the one that we're going to be running today is this run.sh, and we're going to give it our scan date. And you can see that command here, just in case if you guys are following through. So let's go ahead and run that. So you'll notice here that our workflows are now currently running, and our jobs are being completed. So you can see here some terminal output that says job, some job ID, completed, and then it submits a new job, and it executes that job, and it's just this cycle of constant processing. So in this particular case, what we're doing is we're using all of the available cores on our VM and using those to distribute our tasks. Now on an HPC system, this is slightly different because we can use work queue. And what work queue allows you to do is to set up workers on many different nodes that will all connect to the same master node. Now, what this allows you to do, and one of the benefits over this and something like multi-processing or multi-threading, is that you're not limited to the cores contained within one specific machine. Instead, you can basically continue adding more and more workers, and thereby expediting your process as you add more workers. So now you can see all of this print out to the terminal. And what's going on here is we're going through each one of these steps here. So we have done the conversion. We have gone ahead and created our orthomosaic. We just finished clipping to the agricultural plots. And now we're doing the plant detection. So you'll notice here that the printout says faster RCNN. So this is downloading a pre-trained model. And then on top of that pre-trained model, we actually deploy our own trained model on sorghum in this case. And as I mentioned before, we have trained models for many crops. So always feel free to reach out to us if you yourself are growing sorghum or if you have any data that you would like to deploy object detection on. Okay. So I will pause here just momentarily to let you guys catch up and ask to see if there are any questions. So Arian, if there's anything that has come through, please do let me know.
Manuel, someone asked a question about whether Cyverse is accessible from other countries and about the membership and the costs. As far as I know, it's accessible for all the researchers. So if you have any more information on that, it would be awesome.
Definitely. So Cyverse is available to any researcher worldwide. If you use the resources that are that are given by default, you don't have to pay anything. It is a free resource for anyone to use. And a lot of papers that are published use Cyverse because, as I was mentioning earlier, it allows you not only computing resources, but also data storage. And that's really where it shines. So if you're in a different country, you can most definitely use Cyverse. You can also use their computational resources. And if you go to the link that we provided earlier to Cyverse, you can see more information there. And as always, feel free to reach out to us as well.
I have a question about the images used for doing the orthomosaic. Do they have to be in a specific format, specific, like you mentioned that this pipeline can be used also from drones. Do you know if it has been applied, or did you apply it to other, like besides drones? Like what is like are there specific requirements for the images to be merged into the orthomosaic?
Yeah, thank you, Anarita, for that question. And the answer is we have tested our orthomosaicing with other images, such as drone imagery. And the only requirement is that those images be in a TIFF format in order to process it. And Arian is responsible for the orthomosaicing. So Arian, if you want to give a little bit more detail as it relates to that orthomosaicing stuff.
Of course. So, as Manuel said, there are no strict requirements for the images. They can be images from drones, satellite images. Also, we tested the mosaicing algorithm on the handheld camera. I mean, the images that were captured by handheld cameras, and it works with almost all of them. But if your images are geotagged and you have GPS coordinates associated to your images, it's going to increase the accuracy of the mosaicing. So we tested on the drone images that they were associated with GPS coordinates. The gantry images that we have are associated with 4 GPS coordinates representing the corners. That actually helps a lot. And yes, I guess that answered your question.
Yeah, so it's interesting that they also use satellite images. So there are any I don't know, light problems, exposition. So
That's a good question. That's a great question. So many of the image stitching algorithms, they struggle with light conditions and the presence of shadows. But fortunately, our mosaicing algorithm can handle variations in the lighting and shadows. And I believe either Travis or Emmanuel are going to present some results on the mosaicing, and you can clearly see that the shadow is not going to be an issue for the mosaicing.
Yep. So once we set up the other pipeline, we're going to go ahead and continue our presentation in which, as Arian mentioned, we'll go in a little bit more detail for the orthomosaicing.
Okay, cool. Thank you, guys.
Awesome. So now you can see on the terminal, our pipeline has finished processing, and it's telling us to see our outputs in individual thermal out. Now, this is going to be a directory within our computer here. So we can see, if we look into that directory, we can see that there's a individual temps CSV. Now, this CSV is going to contain the temperatures for each individual plant. Now, the way that we do this is, as I mentioned before, we use a faster RCNN detection network. And once we've detected each individual plant, we then use the bounding box area to extract that particular part of the image and then run a k-means clustering algorithm. Now, for those of you that are interested in more details, you can click on this plant temp link here for more information about that particular extractor, as well as all of our code, which is all running open source libraries within Python. Okay, so now that our FLIR thermal data has completed, we will go ahead and click right here below where you see PS2 chlorophyll fluorescence. Now, what we're going to do now is process that PS2 data that Michele was talking about. And similar to before, we've included a figure showing each step of our pipeline. And you'll notice that there are some similarities to the previous pipeline. So we do a conversion to GeoTIFF. We clip to the agricultural plots. In this case, we do image segmentation and fluorescence aggregation. Now, the reason why we don't do individual plant tracking is because we can only collect so many images of this PS2 sensor, because as you will see below, each directory contains 1 metadata file and 102 compressed images. And this is because we want to get an understanding of F0 and FM. So for those of you not familiar with the PS2 sensor, it provides an LED light which saturates the photosystems, and it allows you to determine that F0 and the FM. And from that, you can then calculate FV over FM, which is our photochemical efficiency. This is one of the first indicators of abiotic stress or other stressors, so it is a quite useful trait to track over the entire growing season. So now that we've discussed what the steps are in the pipeline, where you can find our containers, let's go to actually running it. So we're going to go ahead and copy this command into our terminal. So now we have changed directory into our PS2. Here we can see, similar to before, we have our data, which is 2020-0821. We also have our series of shell scripts, and we have this GeoJSON. So for all of our spatial tasks, geospatial tasks, we use a GeoJSON that represents our plot boundaries. And this is how we actually clip to the agricultural plots here. And let's go ahead and now copy the second command here. And actually, before I do that, I want to show you the contents of this particular scan date. So here we only have one subdirectory. And if we ls in here, using this wildcard here, it says ls all of the contents within that directory. It's going to be messy again, so heads up. And you'll see all of those images from 0 all the way to 101. So overall, we have 102 bin files, which are going to be converted into 102 GeoTIFF files for further processing. You can also see here our metadata.json file, which contains all of the information coming off of the actual plant phenotyping platform. Okay, so now that we've looked at the contents, let's go ahead and actually run this. So if we run that command, we are now processing our PS2 data. Now this might take a little while, so now I will ask for Travis to share his screen, and I will go ahead and walk us through some more information about our pipelines as we wait for this to process.
Okay, Travis, you are on.
Awesome. If you could put Emmanuel back on for the talking, for his video. I'm just going to be sharing my screen. He's going to be talking over it.
Oh, so is Michele? I have to...
Emmanuel.
Emmanuel, sorry. I was like, okay, sorry.
Hello, everyone. Okay, so now that our pipelines are processing, let's go ahead and have a conversation about other species that we work with, as well as the orthomosaicing. So here you can see that our modular deployed on multiple crops. So we have processed lettuce data. We have also processed sorghum, which is currently what we're processing. And you can see here, recently, we are now doing some sunflower work as well. So for each one of these species, these pipelines are modular in the sense that they can be used to deploy any species. However, one of the things that is necessary is that trained object detection model. Now this requires labeling, but once you have that model, you can then deploy our object detection and actually individually track your own plants. Now, Travis, if you go to the next slide, we can talk about MegaStitch, which is our orthomosaicing algorithm developed by Arian. So here, one of the problems that we've had with the gantry is inconsistent GPS tagging. And this is due to the fact that as the gantry actually moves, it has error because there's swaying back and forth, or there was wind at the time. All of these environmental parameters ultimately affect the location or the geolocation of our images. And this is very similar to other phenotyping platforms such as drones and other field phenomic processing as well. Now, because of this, naive stitching is not possible, right? It requires photogrammetry, it requires visual features to actually be identified and then matched in order for these images to be stitched together. So if you go to the next slide, Travis. So now we are running geo-correction, and again, Arian has done a lot of work on this, and what's happening here is that we can't use regular orthomosaicing software because of the number of images that we process off of the gantry, as well as the size of those images. So because of this, other orthomosaicing software is not compatible with our data. So we developed Megastitch. And what this does is it allows for large-scale image stitching, and it basically corrects those noisy GPS coordinates. And one of the nice parts is that given your images, we have actually trained a faster RCNN detection network to detect any GCP lid that is a white bucket lid. So if your field contains these types of GCP lids, you can go ahead and deploy that detection model as well. So after we have detected those lids and looked for visual features, we can then actually begin orthomosaicing. So Travis, if you go to the next slide. So let's talk a little bit about the impacts on phenotyping. So as I mentioned before, the commercial software cannot stitch our gantry images. And this is due to low overlap, few distinct visual features, as well as I mentioned, the size and the number of these images. So if this is not addressed, right, accurate plot level orthomosaics cannot be obtained. You need photogrammetry to actually stitch them all together. So as you can see here on the right-hand side of my screen, at the very top, you can see an example that has been passed through Megastitch. And right beneath that, you can see that same region of the field with the raw GPS tags coming off of the gantry. So these images are then analyzed for visual features. They are then stitched together using point matching. And ultimately, those images are resaved and they are used to generate our orthomosaics. Now, this allows us to then move on with our next steps, which are actually clipping to the agricultural plot and detecting our plants. So Travis, if you go to the next slide, this is kind of a general overview looking at a very large region of our field. On the left-hand side of my screen, you can see the before, and on the right-hand side, you can see after. So it provides a significant improvement. You can see here that there's some staggering that becomes pretty apparent. And after passing it through Megastitch, you can see that those plots are then rectified. Okay, so if you can go to the next slide. So this provides a significant contribution to computer vision. And this is something that can not only be used to stitch our gantry images or living room images, but also drone images. And Anarita, this was one of the questions that you had. And this is an example here of some drone imagery that we collected over the same field that the gantry scans, stitched by Megastitch. Okay, so now what we're going to do is check back on our virtual VM. So Anarita, if you can allow for my screen to be shared. Okay. So now we have actually successfully processed our PS2 data. So now let's take a look at some of these outputs. So if we ls that directory, you'll see many outputs. Specifically in this directory, we can see all of the TIFF images that we have just processed. And similar to before, we start with image 0 all the way down to image 100. And this is because we drop image 101. It is not needed. So our output is this 2020-0821 fluorescent aggregation. So if we look there, you can see a single CSV file. And that single CSV file is what provides our FE over FM values. So now that we've taken a look at some of those outputs here, let's now set up a Jupyter notebook and actually start taking a very brief look at the data that we have collected. Now, keep in mind that this is a very small data set that we have used in this case, but we can deploy these systems throughout an entire field and collect many more data points. So now we will give this just a second. So we're going to run this setting up Jupyter Notebook command. So if you hover over and copy this command, paste it into your terminal, and enter your password, this is going to set up an easy Jupyter Notebook. It allows you to then connect to this notebook via your web browser. And once that has completed, we can then click on this URL. So if we click there, one second for it to load. We will see a Jupyter notebook. And this Jupyter notebook is running within our virtual machine. So we now have access to the data that we have just processed. So let's click on this Visualize Data notebook. And what we're going to do here is then go ahead and run some of these commands. So as we were developing this virtual machine, we noted some errors here with this convert v types. So if you just want to go ahead and comment that out, it will solve those problems. And we'll update the image itself, but just keep in mind, go ahead and comment this convert d types for both the FLIR-DF variable as well as the PS2DF. So now let's go ahead and run this Jupyter notebook. So we're going to then click Shift-Enter to run a cell within this Jupyter notebook. And right now we are just importing some of our libraries. We're going to be using Pandas, NumPy, and Seaborn. And let's go ahead and actually run some of our functions. So what this function is doing is it's going to our FLIR-IR directory and pulling that individual temps.csv that we saw on our terminal. And then it's doing something very similar to the PS2DF. You can see here we're going and grabbing that fluorescence aggregation.csv. So let's go ahead and run that, again, using Shift-Enter. And let's go ahead and actually use that function. And if we take a look, we can then see our data frame. So let's take a quick look at that. And here we can see that we have some information listed. So we have which portion of our field it was collected from. We have the species, which in this case is sorghum. We have our irrigation treatments, our range, column, rep. All of this is added during our pipeline. This is information that is contained within that GeoJSON. So during processing, it actually pulls that information and adds it to the CSV. So the most important parts here are this bounding area, this temperature information. So here we provide the median temperature, the mean, the quartile 1, quartile 3. So all this information is stuff that is very important to take a look at as you're automatically processing data, because you don't have very much control during processing. This is all happening automatically. So you want to have a way to catch those instances where you may have an outlier. So as I mentioned before, each one of these rows represents an individual plant. So as you can see here, if we focus in on one of these ranges and columns, you can see we have plant number 1, plant number 2, plant number 3, all contained within the same geographical location. So now let's go ahead and just generate a plot to take a look at our data. And actually, let's go ahead and, so that one, yeah. So we can see the different genotypes there. So it seems like we have an error there. But if you open up your Jupyter notebook, you should be able to see that figure. So that shows you the different genotypes that were analyzed. And basically, you can then use this data frame to then run some more data analyses, so on and so forth. So you can plot not only each individual genotype, but also the treatments that were analyzed using the Seaborn library. Now, in terms of our PS2 data frame, let's go ahead and take a look at that. And here we can see that 3 sections of our field were analyzed. We have 2811, plot 2812, 2813, and we can see the different FE over FM values. Now here we have just simply plotted, bar plot, but contained within our data frame, one of the outputs is actually the number of pixels that were analyzed and some error statistics as well. So keep that in mind. And all of this information is found within that detailed documentation that we have provided a link to. So here we have shown a very small and fast example of our processing pipelines. And keep in mind that this is useful, as we've mentioned before, to not only the data coming off of the gantry, but also to data from other phenotyping platforms. So if you would like to deploy these pipelines, go ahead and follow our GitHub repository. Feel free to clone it. And if you have any questions, always reach out to us at phytooracle@gmail.com, which is provided in the main directory here. So for now, I'm going to ask that Travis Simmons shares his screen again, and I will be talking over to end the presentation. Okay. So in closing, we want to thank the PhenomeForce team for hosting us today. We also want to thank all of the Cyber staff for support with iRods and virtual machine troubleshooting. As I mentioned before, Cyber provides not only data storage and computing resources, but also a lot of help with all of those tools. We want to thank Drs. Duke Pauli, who we are part of his lab, Dr. Cobus Bernard, and Dr. Eric Lyons for all of their support and leadership throughout the development of PhytoOracle. And you can see a picture of all of the team here on the right-hand side of the screen. This has been an effort with a diverse team. For example, we have people in computer science, such as Arian, myself and McKelly, plant science PhD students, Travis, who is an undergraduate student, who's helping with the data science aspect, and as well as engineers. So engineers are necessary to run the gantry because this is a semi-autonomous robot. So us plant scientists cannot do that. So we want to thank you all. And if there are any questions, feel free to ask them in the comments. But this concludes our workshop. So thank you so much for being here today. And thank you for listening in.
Well, thank you, Emmanuel, for this great workshop. So I think it's very interesting because one of the so it's very nice that you are a very diverse group. One of my questions was going to be, what is your background? Because in phenomics, I think it's very interesting that the people with different background, they can collaborate and work together. So you guys are a very good example of this. So I think I'm gonna put all of you guys on so we can thank all of you. So I have a question also about so I'm so sorry that Felipe is not here because he is the one that is always very interested in the orthomosaic, also because he developed the field image R. But my question based on what I do, because sometimes, so I work mostly with fields that they are sometimes not very well, in your case, or most of the time with drone in experimental, but also growing fields, they are in a very specific plot order, so it's kind of also easy to identify the plot carried out and identify the plants. But you also mentioned that this pipeline can also work with satellite images. So for example, I work mostly with trees or with cranberries that, for example, they overlap. And so it's kind of difficult to really define the plot. So my question is do you guys also have experience in this case? Or the plot, the field has to have a specific scheme that you, let's say, you have to provide a specific design of to cut the plot, or the pipeline can do it by itself?
So to answer that, the pipeline can do it by itself because we're using a GeoJSON file, which is very similar to a shapefile for those of you that aren't familiar with it. So as long as that file is provided, that allows you to clip out any plot that you have defined. And then our pipeline just basically iterates through each one of those plot clipped images. And it's agnostic as to how those plots are set up. They're just clipped given that GeoJSON. And you bring up a good point about the detection, right? Sometimes with satellite imagery, that can be extremely difficult due to the low resolution. So in those cases, we also provide containers for plot level averages, right? So we have options not only for individual plant detection, but also plot level data as well. Because in order to do individual plant tracking, it's a lot of work in the field, right? A lot of thinning, making sure that things aren't overlapping very well. So we've tried to make our pipelines as accessible to a wide variety of people, projects, and groups.
Yeah, so I think it's also worth mentioning that if people, like, let's say if one day I need to use your pipeline and need to be adjusted for a specific species, I guess you guys are also willing to work with people that work with different kind of crops. So that's very interesting. Yeah, so thank you so much for having this wonderful workshop. I just want to say that this workshop is still, it's going to be forever on our YouTube channel. So you guys can watch it again. If you have any question, you can reach the speakers anytime. And I'm sure they will be happy to answer any question. So thank you guys for being here today. And so now it's time for me to present I'm gonna share my screen. And so I'm going to introduce the next. So here, I think we didn't update these slides, but there we added a new workshop that will be on June 11th. But for next week, at the same time, same place, we are going to have Jennifer Lin. She is going to talk about Arduino, which is another small computer that can be used for very low-cost phenotyping platforms. She's going to show how to use Arduino for obtaining long-term pH reading in plants tissue. So if you register for this workshop, you don't need to register again. You are already in our mailing list. And so we are going to send information about the workshop on Thursday, as we do for our workshop. But if you think that this workshop can be of interest for any of your friends or colleagues, please share the link with them. And with that, I'm gonna thank you all for being here today, and see you next Friday. Bye-bye.